There you go.
Okay, during the third stage of mitosis, which is anaphase, the chromatids are pulled apart, as you see here, creating two identical chromosomes, which are held by the microtubules here, or arms of the centrioles, towards each pole of the cell, which makes way to the cycle of telophase here. As telophase happens, the chromosomes arrive at the poles and are relaxing, while the arms of the centrioles begins to dwindle, and they recycle the parts from the arms of the centrioles into the cytoskeleton, which then the nuclear envelope reappears along with the nucleoli, which makes way for cytokinesis. And it starts to build a furrow here in between. Well, cytokinesis happens. It's a process at the end of mitosis, and is a method where the plasma membrane pinches the cytoplasm to create two new cells. It's like elastic on a balloon. And so it pinches off to make two complete packages of cells. There you go. And that's the end of cytokinesis. That's your lesson for the day.
Hi, my name is Nita Thomas, and me and my group worked on this diagram to present it with you to show you what each steps are in the cell cycle. Cell cycle number 1 is called the G1. The G1 phase mainly includes the growth of the cell. The cell is in its smallest size at the beginning of G1. There is also an opening before the cell hits G1, and that's called GO, and that's where the cell's sent to go to GO phase when cells don't divide properly. And then the cell carries on, and then there's another cell check for at the end of stage G1, which that cell check is cell size adequate growth, to make sure the cells are growing to the right stage. And the next stage in the cell cycle is the S phase, which is the period in which chromosomes are replicated. The chromosomes consist of two sister chromatids, the S phase. And then the cell cycle slowly goes through the interphase, and then there's another checkpoint at the end of the S phase, and this checkpoint checks for is the DNA is photocopied, meaning it is exactly the same as the first cell from G1 where it's replicated. And the third stage is G2, which is when the cell prepares for division of its nucleus. A protein that makes microtubules for cell division is synthesized. The cell also takes inventory and makes sure it is ready to continue with mitosis. This is the G2 phase. And there's also another checkpoint as you follow along the interphase cycle, and this checkpoint is for cells to prepare for mitosis, and then you enter the mitosis phase. Right now we're only worrying about the G1, the S phase, and the G2. And this is the presentation that we presented you with. So enjoy.